In this video, we will be discussing about non-Mendelian inheritance or deviation from Mendelian inheritance. There are certain phenomena such as incomplete dominance, co-dominance, multiple allelism, polygenic inheritance and pleiotropy which do not show Mendelian ratio. First, we will take up incomplete dominance. Incomplete dominance, in this, the dominant character is not completely expressing itself in heterozygous form. It is inheritance in which offspring shows characteristics intermediate between two extreme parental characteristics. In Mendelian ratio, in heterozygous form, or the F1 of monohybrid cross showed complete dominance of the dominant character. For example, if we cross tall and short plants, in F1 generation and heterozygous form, we got all tall plants. But in this case, the flower color in snapdragon or dog flower, that is Antirrhinum species and Mirabilis jalapa, we can see that heterozygous form is having an intermediate characteristics. For example, if the red flower, which is having dominant genes capital R and a white flower, recessive character small r and small r, when they are crossed, the intermediate character pink was obtained in F1 generation and it is small r and capital R as the heterozygous form, and this means that the complete capital R is not completely dominant over small r. And this is a deviation from Mendelian ratio because the phenotypic ratio in F2 generation is 1:2:1 instead of 3:1 because heterozygous form showed pink color rather than red color. And genotype ratio is also 1:2:1 and phenotype ratio is also 1:2:1. Now if we see the co-dominance. Co-dominance means together the genes are dominant, and it is the inheritance in which both alleles of the gene are expressed in the hybrid. Both the alleles are expressed in the hybrid. An example is ABO blood grouping in humans. ABO blood groups are controlled by gene I. This is important that gene I controls the blood groups, and there is a production of sugar polymers known as antigens which protrudes from the plasma membrane of RBC. You are seeing here protrusions of the antigens from the RBCs. And if the antigen is A, the blood is A. If antigen is B, the blood group is B. The gene I has three alleles, IA, IB and small I. IA and IB, they produce slightly different type of sugar. You just see here it is depicted by round and here it is conical. Whereas small I does not produce any sugar. In this case, we are having both A and B. That means this blood group is AB. Here both the type of antigens are found. So, if all these two groups A and B are present, then both are expressed in the blood group. You see, if father and mother, if father is IA and small I. That means it is recessive, and mother is IB and small I. That means small I is recessive and it is carrying the antigen B. If they are crossed, we will be getting genotypes as IAA or IBB, IAB or small I in this manner. So all the different types of blood groups we will find here. Allele from parent 1 is A, from 2 is A, then offspring will have A as the allele and the blood type will be A. If allele is A and second parent gives B allele, then the blood group is AB. Similarly, if A is from parent 1 and small I from parent 2, then we are having A blood group because I is recessive and A is dominant, so it is expressed. But if when allele from parent 1 is B, allele from parent 2 is A, that means both the alleles are present and both are expressed. So here we are getting AB blood group again. And if the B is the allele from parent 1, B from parent 2, we are getting B blood group. Whereas if parent 1 and parent 2 contribute both small I, then we are having O blood group done because they are recessive, and this O is expressed in the homozygous form. And when A and B, they are present together, that is important. They both express their own types of sugar, and this is due to co-dominance. Now we will see another important Mendel's deviation, that is multiple allelism. Multiple allelism, it is the condition in which more than two alleles govern the same character. An example is ABO blood grouping that we discussed. Here three alleles are controlling the same character, that is IA, IB and small I. So it is multiple allelism. Next is polygenic inheritance. Polygenic inheritance, in this the traits are controlled by three or more genes, multiple genes. There the gene was same but alleles were different. Here we are having different genes. For example, human skin color, human height, and it is also influenced by environment. And the phenotype is the contribution of each allele. Effect of each allele is additive. So this skin color is controlled by three genes, capital A, capital B, capital C, and dominant capital A, capital B, capital C responsible for darker skin, and recessive small a, small b and small c for light skin in color. So if all the genotypes with all the dominant alleles are there, we have darkest, and if genotype with all the recessive alleles are there, we have lightest skin color. And genotype with three dominant alleles plus three recessive alleles gives the intermediate skin color. Thus, number of each type of alleles in the genotype determine the darkness or lightness of the skin. Our last deviation is pleiotropy. Pleiotropy, single gene exhibits multiple phenotypic expression, and such a gene is known as pleiotropic gene. And this gene affects the metabolic pathway which contributes towards different phenotypes. For example, star synthesis in P, phenylketonuria, sickle cell anemia. These are some of the examples of pleiotropy. Phenylketonuria and sickle cell anemia that we are going to discuss in our later videos. And in this, the mutant gene exhibit many phenotypic effects. such as in phenylketonuria, we have mental retardation, reduction in hair and skin pigmentation. In case of star synthesis in P, it is controlled by gene B, and the capital form or dominant form produces large size and more starch, and the recessive form produces less starch and small size seeds. Whereas when both are crossed, they produce intermediate size of seeds. So in this video,